Hello, my name is Manohar Mulchandani, and I'm going to talk about transcriptome assembly and some specific bioinformatic considerations related to that. We begin with some background slides that will explain what the transcriptome is and the need for assembling the transcriptome. We will then talk briefly about the RNA-Seq method for transcriptome assembly, which uses the next generation sequencing technologies to generate short reads from the transcriptome. Finally, we will talk about how the transcriptome is assembled from the short reads and the related bioinformatic considerations. So we start with the central dogma that we are all familiar with. According to this dogma, DNA is transcribed into RNA, and then the RNA is translated into proteins. Now, there are various types of RNA, and one of them is called the messenger RNA, commonly referred to as mRNA, and that is the focus of this presentation. The mRNA is transcribed from protein encoding genes in the human genome, and after transcription, it undergoes some pre-processing, and then it leaves the nucleus and enters the cytoplasm where it is translated into proteins by the ribosomal machinery. On the right is the formal definition of the transcriptome from Wikipedia. As is stated there, the transcriptome is comprised of all the different types of RNA that are transcribed from the DNA. Note that not all RNA is translated into proteins. Some of these RNA perform other functions in the human cell. Note also that the transcriptome varies across different cell types. Let's go a bit deeper to understand how the transcription process creates the mRNA. As you can see in the central panel where the red oval is, the transcription process actually creates what's called a pre-mRNA. This pre-mRNA is spliced to produce what's called the mature mRNA. It is this mature mRNA that is exported to the cytoplasm and is translated into proteins. Now, this splicing process actually adds variety to the rather drab and boring central dogma and makes life for the bioinformatician difficult or interesting depending on one's perspective. The human body manufactures over 100,000 proteins, even though there are only about 20,000 protein encoding genes in the human cell. Splicing causes this to happen, and the figure on the right shows a little more about how that happens. As we all know, proteins are encoded by genes, and the gene transcription process eventually produces the mature mRNA. However, the protein encoding region of the gene is not contiguous and is punctuated by what are called introns. In other words, the gene is actually made up of different regions called the exons and the introns. It is the exons that carry the information to encode for the proteins. In the picture on the far right, the exons are shown as boxes, while introns are indicated by thick lines. This is just a distinction in the drawing. In the actual gene, one doesn't see this difference, and neither does the transcription machinery. So the transcription process ends up transcribing both the exons and the introns. Clearly, we need to get rid of the introns, and that is done by the splicing process. The splicing process removes the introns from the pre-mRNA and creates the mature mRNA. But we are not done yet. Occasionally, the splicing process will also remove certain exons, resulting in several different types of mRNA molecules coming from the same gene. Each different form of protein is called an isoform. Thus, one gene can result in different types of mature mRNA, thus enabling the creation of different proteins. In the diagram, we see two types of combinations of exons, but there could be other types. Note that non-adjacent exons in the gene can end up being adjacent to each other in the mature mRNA. The end result is that when studying the transcriptome, we not only want to identify which genes are expressed, but we are also interested in the different alternative splicings of the pre-mRNA that result in multiple types of mature mRNA from the single gene. Next, let's understand why transcriptome assembly is helpful and how it is done. So why do we need to assemble the transcriptome? Well, among other things, a study of transcriptome helps to understand the function of genes. It also helps us understand changes in gene expression that happen in a disease cell so that we can treat them. The link that I've provided provides more information about this, as well as information on methods used for transcriptome assembly. Uh, in this session, I'm going to talk about a method referred to as RNA-Seq. This is a relatively recent method that's based on the next generation sequencing technologies. RNA-Seq has several advantages, such as the ability to not only detect a transcript, but also estimate the abundance of the transcript. It also has a higher dynamic range, which enables it to better detect transcripts that have low abundance. Its primary disadvantage is that it generates very short reads, often with errors, and this requires considerable bioinformatics effort. For the transcriptome assembly, the RNA-seq method makes use of the reference genome. To that end, the mRNA is first reverse transcribed to form a DNA. The DNA so generated is called complementary DNA or cDNA. cDNA constitutes the same nucleotides that were in the gene from which the RNA was first transcribed, albeit with one important difference. Since the introns have been spliced out of the mRNA, the cDNA doesn't contain any introns. So one cannot directly map the cDNA to the genome to infer the transcriptome, and we need another way to accomplish this. So how does this mapping happen? Well, actually, the cDNA is shredded into smaller fragments that can be sequenced by the NGS technologies, resulting in short reads. The shredding process is very similar to the newspaper explosion process. The main difference is that instead of exploding hundreds of thousands of copies of New York Times of a particular day, here we are exploding hundreds and thousands of copies of many different newspapers on the same day, since we have many different types of mRNA transcripts. The short sequence reads are mapped to the reference genome, and we'll discuss this in the next slide. We'll first summarize the overall process. So we have a gene say with 4 exons, which is transcribed into a pre-mRNA, which is then spliced out into a mature mRNA. This is where the RNA-seq process kicks in. It first reverse transcribes the mature mRNA into cDNA. The cDNA is then shredded into sizes that can be sequenced on an NGS platform. Now the shredded cDNA is then mapped to the reference genome, and it is this mapping that reveals the transcriptome to us. Note that the shredding of cDNA is random, and so we cannot guarantee that each cDNA piece maps to only one exon. In this diagram, there are 2 cDNA fragments, each having portions of 2 exons. In a large-scale study, the numbers are much higher. Also note that cDNA fragment may not necessarily contain fragments from adjacent exons. In case an exon was spliced out during the splicing process, we may have cDNA fragments that have portions from exons that are further apart. Now, in this diagram, the coloring scheme and the black boundary are only to illustrate how exon fragments may be combined in a cDNA fragment. In real life, we do not have information about which exon paths are combined in a cDNA fragment, and neither do we have information about the boundaries. In addition, we may not always have information about all exon-intron junctions in the genome. So we may first have to identify potential exon-intron junctions, which can help identify potential exon-exon combinations. Now, exon-intron boundaries have a typical signature, and although there are variations, it is possible to come up with potential intron-exon junction. And as of today, we have information about many existing such junctions which we can leverage. Now, assuming that we have information about all the exon-intron junctions, we can begin the process of cDNA mapping. For each cDNA fragment that doesn't map completely to an exon, we try to match it to all possible exon-exon combinations. Since we do not know how the cDNA fragment is constituted, we split the cDNA fragment into 2 at each nucleotide position in the fragment, and then try to match the 2 paths to 2 adjacent or neighboring exons. Now this is a slightly simplified explanation, but that's how it happens. And the challenge is to be able to do this fast, because we're talking about millions and millions of such mappings that we need to do. And this is what requires efficient algorithms. Having done the programming exercises, I'm sure we can all appreciate the complexity of this task. And the way I have laid it out, it perhaps makes it sound very simple. but in real life there are other problems. For example, errors that complicate the issue. Uh, that's all I wanted to talk about on this subject. On the next slide, I have provided a reference to a review article that summarizes the state of the art of RNA-Seq as in 2010. Uh, I think it's still a good read for those who are interested.